Hello friends, welcome back. So in today's session we'll be learning about nerve muscle physiology, one of the very interesting topic in physiology and forms the basis of many physiological processes in systemic physiology like CVS, CNS and even respiratory system. Now let us begin with what are the topics we'll be discussing in nerve muscle physiology. The first thing we'll be learning in nerve muscle physiology is about the membrane potentials, that is the resting membrane potential and action potential. These two forms the very basis of all the impulses which travel across the nerve fiber or the muscle fiber or various receptors. So resting membrane potential and action potential is the first thing we'll be discussing in nerve muscle physiology. Next we'll be discussing is about the neuron or the nerve fiber, because nerve muscle physiology is about the learning of nerve and a muscle. So the second important thing will be about the neuron or the nerve fiber, because nerve fiber forms an integral part for the study of nerve muscle physiology. Then we'll be discussing about the classification of these nerve fibers as well as the properties of these nerve fibers. All this will be included in the nerve fiber and its further topics. We'll be also going to discuss the point where the nerve fiber is connected to the muscle fiber. It is called as neuromuscular junction, one of the very sought after topic for every individual in nerve muscle physiology. It's neuromuscular junction. Neuromuscular junction forms the one of the very integral part of nerve muscle physiology. After neuromuscular junction, we'll be discussing about the mechanism which leads to muscle contraction. See, nerve muscle physiology is all about what? It's about the impulse which is in the neuron which travels across the nerve fiber, and this impulse which travels from the neuron to the nerve fiber and it goes to the muscle or the muscle fiber, it causes the muscle fiber to receive that impulse, and when the muscle fiber receives the impulse it contracts. So how this NMJ leads to the excitation of the muscle fiber, and when the muscle fiber is excited, how it contracts. So it comes under one of the very interesting topic, and that is excitation contraction coupling. Excitation contraction coupling. So where it's a point where the excitation of the muscle fiber is converted to the contraction of the muscle fiber. So excitation contraction coupling. Next, after excitation contraction coupling, we'll be discussing the next topic, and that is the mechanism of muscle contraction, or you can say it molecular basis of muscle contraction. So mechanism, or you can say molecular basis of muscle contraction. Molecular basis of muscle contraction, where we will try to understand the molecular structure of the actin, molecular structure of the myosin, and how do they help or how do they interact for contraction. So mechanism or molecular basis of muscle contraction. Next, we'll be discussing about the certain properties of muscle contraction. These are the topics which forms our principle for CVS and respiratory system, which called as properties of muscle contraction. Properties of muscle contraction. Like, let me give you an example. It's like isometric or isotonic contractions, preload and afterload in the muscle, Frank-Starling principle working on the muscle. So these are topics we'll be discussing in the properties of muscle contraction. Later on, we'll be discussing about certain applied physiology in muscle contraction. Applied physiology for the contraction of the muscle fiber. Then, apart from this, we'll be discussing certain few topics like how this contraction process occurs in smooth muscles or in cardiac muscles. Smooth muscles and cardiac muscle. So these are topics we'll be discussing in all over our nerve muscle physiology. Let us just recall what we'll be discussing in nerve muscle physiology. We'll be starting with membrane potentials, that is resting membrane potential and action potential. Then we'll be moving on to the nerve fiber itself, how the nerve fiber works, what are the properties of nerve fiber, how do we classify nerve fiber, certain points like degeneration and regeneration of the nerve fiber. Then we'll be going for the neuromuscular junction itself, how does the nerve transmit the impulse to the muscle, and when the muscle receives the impulse it goes under excitation and contraction. So we'll be discussing excitation, contraction, coupling. Later on we'll be discussing the molecular basis of muscle contraction, then certain properties of muscle contraction, applied physiology of nerve muscle system, and certain various mechanisms which are involved in smooth muscle contraction and cardiac muscle contraction. Now let us begin with our first part, that is membrane potential. So what we'll be discussing in membrane potential, and what is membrane potential itself? Membrane potential is a chapter topic where we discuss about the voltage. Potential is all about voltage. So voltage differences which exist inside the cell and outside the cell, and how these voltage differences they change under various conditions, and how the resting membrane potential has originated, and how this RMP changes to action membrane potential or action potential on receiving an impulse, sudden impulse. So membrane potential is a topic which encompasses your resting membrane potential and action potential. First, resting membrane potential. Now, RMP, whenever you get this word resting membrane potential, remember it's a condition which exists in the cells which are excitable cells. In our body, we can classify the cells under two types, an excitable cells and a non-excitable cells. The excitable cells are the cells which exist in two states, a resting and an excited. Say for example a nerve or a neuron. So neuron, when it is not transmitting an impulse, when it's not transmitting an electrical impulse, that neuron is resting, and hence the neuron is an excitable tissue which can exist in two states, a resting state and an excitable state. In the resting state, it establishes resting membrane potential, and when excited, it produces action potential. So an excitable cell is a cell which is capable of changing its potential, and hence for excitable cell we can use the word RMP. Now for the non-excitable cells, for example RBCs, red blood cells. The red blood cells, they have resting membrane potential, but their resting state does not change. They don't generate action potential. So for them, the resting membrane potential can also be known as just membrane potential. So remember, membrane potential word can be used for the cells which are non-excitable, RBCs, certain epithelial cells which are non-excitable cells, hepatic cells. The cells which cannot excite, which cannot change their potential, are called as non-excitable cells. Where the cells which can change their potential, they are called as excitable cells, and for them we can use the word RMP. So remember, RMP is a word which can be used typically for the excitable cells, and that's what we are going to focus on for our understanding purposes. The excitable cells are the cells which are capable of changing their potential from resting to excited state, and hence for them we use resting membrane potential. Now what is the voltage, what is the potential of RMP, because it's resting membrane potential. So as a general term, remember, as a general rule, always remember all the excitable cells and even all the non-excitable cells, they always maintain a negative voltage inside them as compared to outside. A resting membrane potential is a potential in which the cell maintains a negative voltage inside its membrane. Remember, a negative voltage is maintained inside the membrane is said to be the resting membrane potential, RMP. Fine. Now you must be confused, or you must be having a question that if inside is negative, do we consider outside positive, or what else do we consider outside? Now here there is one more concept. When we use the word resting membrane potential, we are concerned about what is the voltage inside the cell. We are not concerned what is the voltage outside the cell. Means for RMP, we always remember, for RMP we always consider outside voltage as 0 millivolt. Whatever with outside voltage, we consider it as 0, and we compare the voltage inside the cell as compared to outside, which is considered 0. How? Let us take a very simple experimentation here. We have two electrodes in our hand. We place one electrode inside the membrane and we will place one electrode outside the membrane. And these two electrodes will be connected to a voltmeter. Now, when you connect these two electrodes to a voltmeter, the voltmeter will give the difference across the two membranes, two sides of the membrane. And it might show some voltage. But with some instrumentation, with some mechanical instruments, we will make outside electrode such a way as that we make that electrode a electrode which is neutral, or we can say electrode which gives you a 0 voltage. So whatever be the outside voltage, we will not consider that. We will nullify it and will bring that voltage to 0, and then we will see what is the difference now. And whatever difference you observe at this point, that difference will be the difference of RMP. So RMP is a voltage which is inside the cell. Resting membrane potential is a potential which is inside the cell. It is always negative, and in this condition, in RMP, we consider outside voltage as 0 millivolts. Whatever may be the outside voltage, plus 10, plus 20, plus 30, we are not concerned. We consider outside voltage 0. So if I say that RMP is minus 70 millivolt, just an example, then this minus 70 millivolt is inside the membrane, and outside the membrane, the voltage is 0. In this way, we have to consider resting membrane potential. Now, there is always certain exceptions. There is one more exception here, and that is if we use the word transmembrane gradient. Now, transmembrane gradient is quite different from RMP. Now, what is his? Now, see, if I use the word transmembrane gradient, if I use the word transmembrane gradient, then the situation changes quite a bit, and RMP here is also going to be different. How? Let us see. Say, for example, a cell, a neuronal cell or any cell, has some negative voltage inside it and a positive voltage outside. Say, outside we have plus 30 millivolts and inside we have minus 70 millivolts. Now, in such situation, if I use the word transmembrane gradient, then what will be the actual difference between these two voltage? In such condition, the difference will be, remember, the difference will be 100 millivolts, because here the voltage is gradient from plus 30 to minus 70. So when we use the word transmembrane gradient, we are using the voltage difference across the two sides of the membrane. So we consider outside voltage in this case, and here it becomes plus 30 outside and minus 70 inside. So the overall difference from plus 30 to minus 70 will be 100 millivolts. So in this case, the transmembrane gradient is 100 millivolts here. we have one question here, and I will ask the question right now and I'll give the answer at the end of this concept. The question is, which cell which cell in our body has highest transmembrane gradient? And the options are the endocochlear cells, the gustatory cells, the olfactory cells, or the touch receptors. Which of these cells have highest transmembrane gradient? I repeat, highest transmembrane gradient. Endocochlear cells, olfactory cells, gustatory cells, or touch receptors. Which of these cell will have a highest transmembrane gradient? Think of the answer. I'll just continue and then at the end of this point, I will give you the answer. Now, in transmembrane gradient, we consider the voltage difference across the two membranes. What if I ask the RMP right now? For this cell, if I ask what is RMP? Now, what do you think will be the RMP here? What will be the RMP or resting membrane potential in this case? As we have considered in our previous part, for RMP, whatever may be the voltage outside, we always consider that voltage as 0 millivolt. We always consider that voltage as 0 millivolts. So RMP for the same cell becomes minus 70 millivolts. So remember, these are the two very interesting terminologies here, a RMP and a transmembrane gradient. So if you are given a question like what is RMP for the given cell and you are given various voltages, remember to consider only with the voltage inside the membrane. Don't take any mechanical or any mathematical difference. Just consider the voltage inside the membrane. So in this case, the RMP is still minus 70. But if you are asked about transmembrane gradient, then you have to consider the voltage on either side of the membrane, and that will be the overall difference, that is the magnitude of difference between the two values. Magnitude of difference, it's not mathematical difference, it's a magnitude of difference. So from minus 30 to minus 70, it will be almost 100 millivolts here. If you remember, we used to have certain integer scales like this, and we used to have 0, and we used to go minus 10, minus 20, minus 30, minus 40, plus 10, plus 20, plus 30, plus 40. So in this way, we go from minus 30 to minus 70, and the overall difference becomes 100 millivolts. So that's how you have to imagine the changes in the voltage here. So this is what I wanted to start with the RMP. Transmembrane gradient and resting membrane potential are two very, very important terminologies in terms of membrane potential. RMP is always used for excitable cells. In transmembrane gradient, we use the voltage, or we consider the voltage on either side of the membrane. And for non-excitable cell, the word membrane potential can also be enough. Now, what is the answer? I have asked the question, which cell has highest transmembrane gradient? So remember, the answer is, I'll just again wait for a minute. Let me draw the diagram so that I'll give you an idea here. In this cell, the outside voltage is plus 80 millivolt, and inner voltage is minus 70 millivolt. So in this case, the transmembrane gradient is of 150 millivolts. And which cell is this? Remember, this cell is endocochlear cell. The endocochlear cells has highest transmembrane gradient. Endocochlear cells are a very typical exception here. Endocochlear cells in our body, they have highest transmembrane gradient, means although their inner voltage, their RMP is minus 70, but outside the fluid outside the cell is actually so much positive that it has plus 80 millivolt. And for this case, we do have to consider the transmembrane gradient rather than just considering the RMP. So remember, this is one of the very important MCQ also for you. The endocochlear cell has highest transmembrane gradient. Now, let us move to the origin, that how do we have a minus 70 millivolt for RMP? And does all the cells have same minus 70 millivolt? The answer is no. Various cells have various RMP values. Let us write down few values for few other cells. For example, for a neuronal cells, the RMP is minus 70 millivolts. RMP is minus 70 millivolts. For skeletal muscles, remember the RMP is minus 90 millivolts. For neuronal cell, it is minus 70 millivolt. For skeletal muscle, it is minus 90 millivolts. For cardiac muscles, that is atrial and ventricular muscle fibers, for cardiac muscles, it is minus 89 to minus 90 millivolts. Whereas for smooth muscles, it is quite different here. For smooth muscles, it is minus 45 to minus 50 millivolts. Minus 45 to minus 50 millivolts. Only for this case, remember, for smooth muscle cell, the RMP is not fixed. It is quite changing. Minus 45 to minus 50 millivolt. And hence it is also called as basal electrical rhythm. BER means even at rest, the smooth muscles maintain a rhythm of RMP, that is changing RMP minus 45 to minus 50, and hence it is called as the basal electrical rhythm. Then for pacemaker cells, for example SA node and EV node, what will be the RMP here? For pacemaker cell, the RMP is minus 65 millivolts, always as some books mentioned as minus 60 millivolts. So it is something around minus 60 to minus 65 millivolts. Remember, for pacemaker cell, the RMP is minus 65 millivolts, or in some cases it is also minus 60 millivolts. Then what will be the RMP of, say, RBC, where we can call it as membrane potential RMP for RBC, that is we call it as membrane potential and not actual RMP, but it is only membrane potential. It is minus 12 millivolts. So for RBC it is minus 12 millivolts. Because this voltage will not change, it can also be called as membrane potential. Okay, so RMP is not fixed. RMP is not fixed for all the cell. It is not common for all the cells. It differs from cell to cell. We have given few examples here, and I wish if you could remember these examples, because these values might help you to remember or understand future topics. For neural cell, it is minus 70. For skeletal muscle, it is minus 90. For cardiac muscle cell, it is minus 89. For smooth muscle cell, it is minus 45 to minus 50. For pacemaker cell, it is minus 65. And for the RBC, which is a membrane potential, it is minus 12 millivolts. So we can just generalize, make a statement that RMP in our body, RMP in human body, it has got a range from minus 5 to minus 100 millivolts. Just to give an idea, we have a range of cells and every cell has different RMP values. So we can have a range of RMP in our body which can range from minus 5 to minus 100 millivolts depending upon the cell type and the environment surrounding the cell. Now, let us write down few peculiar features of resting membrane potential. RMP or resting membrane potential, remember, it is also known as a steady state potential. Steady state potential. It means unless and otherwise the cells are excited, the cell maintains this potential throughout their cycle. Means if the cell is not excited, it will maintain the resting state throughout till it's getting excitation. So it's a steady state potential. The cell maintains RMP. Remember, the cell maintains RMP to a specific value. And when we maintain RMP to a specific value, it is a part of homeostasis. It maintains RMP. The cell maintains RMP. They are not by default. The cell have to work to maintain that value. Number 3, because they maintain RMP, they need to maintain the RMP they use energy. So it's an energy requiring process. It's an energy requiring process. It is not free of cost. Whenever you have steady state matter or steady state activity, to maintain the steady state, you require energy. Like homeostasis itself is an energy requiring process, because homeostasis is a steady state activity. We maintain temperature at 37 degree, we maintain blood pressure at 120/80, we maintain pH at 7.4. So these are the steady state. So any place, anywhere, wherever we maintain a steady state, we require energy. So it's an energy required process. So RMP requires energy. Always remember, RMP requires energy. And fourth point, which goes in favor of RMP, it's a homeostasis of the cell. You can say it's a homeostatic mechanism for the cell. The cell has a homeostatic mechanism for RMP because it maintains RMP. Whatever may the changes outside the cell, it just tries to maintain that steady state potential, minus 70 or minus 90 or minus 89 or minus 65, irrespective of the changes outside the cell. So remember this very important features of RMP. It is a steady state potential. It is maintained. The RMP is maintained. It is not by default. The cell need to maintain RMP by investing certain energy in form of ATP, and it's one of the homeostatic mechanism of the cell. These are the four very interesting features of RMP. Now let us go with the concept how RMP is generated. All of us needs to understand this physiology. Very important, that how a cell develops a negative voltage inside it and how it maintains that negative voltage inside it. Because as a clinical point of view, there are certain conditions which are typically based on this concept of RMP. Let me give two names here, two terms here, two conditions. One condition is known as the hyperkalemic periodic paralysis. Remember, hyperkalemic periodic paralysis or hypokalemic periodic paralysis. The condition which is either because of increase in potassium ion concentration, or the condition which is because of decrease in potassium ion concentration, which causes an attack of periodic paralysis, are typically based on the concept of RMP. Or the hypocalcemic tetany, or other conditions where the cell becomes more excitable or the cell becomes less excitable. They are all based on the concept of RMP. So to understand those clinical conditions, we need to understand the concept of genesis of RMP. How RMP is generated. Genesis of RMP. Remember, RMP is generated. It is not by default. The RMP is generated and maintained inside the cell. So to generate RMP, remember, we require three important factors. There are three important factors which are responsible for the generation of RMP. Number one, we will look for the concentration gradient of the ions across the membrane. The first factor is concentration gradient. Concentration gradient of the ions across the membrane. Number two, apart from concentration gradient, we'll be looking for the permeability of the ions across the membrane. Permeability, remember, after concentration gradient, we'll be looking for the permeability of the ions across the membrane. So concentration gradient, permeability are the two very important factors, first two factors. And the third factor, yes, which is inseparable and which plays a very key role here, is sodium potassium pump. The activity of sodium potassium pump also plays a crucial role in the maintenance of RMP, generation and maintenance of RMP. So remember, there are three important factors which play a very crucial role in generation of RMP. Number one, concentration gradient of the ions across the membrane, because ions do move. Sodium ions, potassium ion, chlorine ions, they do move across the membrane, and their movement will determine the voltage inside the membrane. Permeability, how much an ion moves in how much quantity, what is the rate of movement, that determines the RMP. And the third one is sodium potassium pump. So these three factors very important to determine the potential inside the membrane. Let us go with one example here. Say we'll take an example of a nerve cell which has a minus 70 millivolt. So I'll just draw a cell over here, and we will write down those three ions which primarily determine the RMP. So we have a ECF compartment, we have an ICF compartment, and in these two compartments the cells are distributed. Which two cells? Remember, we are talking about RMP, and the cells we're talking about is the distribution of ions we're talking about is ions in ECF and ions in ICF. So three ions. One is sodium ion. Now I would like to know from your side, in which side of the cell we have sodium ion in higher concentration, in ECF or in ICF? The answer is sodium ion is more in the ECF. Sodium ion is more in the ECF. Number two, potassium ion. Where do we have potassium ion in higher quantity or in higher concentration? It is inside the cell. Potassium ion is in higher concentration inside the cell. And the third ion is chlorine ion. Where do we have chlorine ion in higher concentration? Outside the cell or inside the cell? The answer is outside the cell. The chlorine ions are in higher concentration outside the cell. So why do we have this differential distribution? It is because of one of the principle. And what is that principle? It's a principle of the Donan membrane equilibrium. Remember, the RMP is generated because of the movement of the ions, and this differential distribution of the ion is based on a concept of Donan membrane equilibrium. What does Donan membrane equilibrium says? The Donan membrane equilibrium says that inside the cell there are certain larger molecules like proteins which cannot move outside the cell. And these proteins, because they cannot move outside the cells, they affect the movement of the ions across the membrane, which can move across the membrane. So the Donan membrane equilibrium is about the differential distribution of the ions across the membrane, which can diffuse in the presence of the molecules which cannot diffuse and which have certain charge on them. Okay, so proteins have a negative charge, and this negative charge on the protein, it affects the distribution of the sodium and potassium ion or even chlorine ion. Therefore, the overall distribution of the ions across the membrane is based on a concept of Donan membrane equilibrium. So we will add one point in our previous features, and the fifth one is RMP, you can say, is also because of Donan membrane equilibrium. So you can have one more feature of RMP. The Donan membrane equilibrium is one of the concept which also helps in determining the RMP. That's why the cells are always negative. You can see, because proteins are larger molecules, they cannot cross the cell membrane directly, easily, and hence they stay inside the cell. And because they remain inside the cell, their negative charge contributes to negative environment inside the cell. So Donan membrane equilibrium gives rise to this sodium and potassium distribution. Now, since we are considering these three ions, remember sodium, potassium and chlorine. These three are the primary ions which determine the RMP of any cell. Remember, sodium, potassium and chlorine. How? We will discuss these three ions on the basis of these 2 points, that is concentration gradient and permeability. So let us first go with concentration gradient. So see, if I say that the cell is at rest and the cell has a minus 70 millivolt. How much? Minus 70 millivolt inside it. So with respect to concentration gradient, now let us see. Suppose if sodium ion is more outside, and by diffusion sodium ion is going to move from where to where? By diffusion sodium ion is going to move from outside to inside. Sodium ion is going to move from outside to inside. So if by concentration gradient we have sodium channels which allow sodium ions, and if sodium ion is going to diffuse from outside to inside, what voltage sodium ion is going to brew inside the cell? If sodium ion is going to move from outside to inside, what voltage or what charge sodium ion will bring inside the cell? Definitely it is going to bring positive voltage. So the question is the sodium ion concentration gradient can fully be responsible for RMP generation? No, we want negative voltage and sodium ion movement is giving us positive voltage. So sodium ion movement as of now, or sodium ion concentration gradient as of now, is of not much useful to us. In a very simple way, remember this thing. Sodium ion is a positive ion, it's a cation, and it's more outside the cell. So if sodium ion is going to move from outside to inside, it is going to bring positive voltage inside the cell. And because it's bringing positive voltage inside the cell, sodium ion movement cannot help us to generate RMP. On the other hand, let us see what do we have for potassium ion. What do we have for potassium ion? If potassium ion is more inside the cell, it's a positive ion and it's going to diffuse now. So in which direction the potassium ion is going to diffuse? Potassium ion will diffuse from inside the cell to outside the cell. And we are concerned about the changes inside the cell. So if potassium is a positive ion and is going outside the cell, inside to outside, potassium ion is going to diffuse from inside interior of the cell to outside or exterior of the cell. What charge is going from inside to outside? Positive charge. The positive charge are moving out. If positive charges are moving out, what happens inside? What happens in the interior? In the interior of the membrane, if the potassium ion are being lost or they are going out, if positive going out, what remains inside is negative voltage. That's the law of the physics. If we are losing a positive, there is no positive, it means negative. So movement of potassium ion generates a negative voltage inside the membrane, and yes, we can say that potassium ion movement to a very greater extent is going to help us for RMP. So yes, potassium ion is going to be a hero for us as of now. Sodium ion, not that important hero. Potassium ion, yes, it's a hero for us because it's going to generate a negative voltage inside the cell. And what about chlorine ions? Chlorine ions are going to move from where to where? They are going to move from outside to inside. And when they are moving from outside to inside, what charge are they going to bring from outside to inside? Remember, chlorine ions have a negative charge. They are anions. So when chlorine ion moves, they will bring a negative charge inside the membrane, and because they are bringing a negative charge inside the membrane, chlorine ion is also to a certain extent is going to help us to generate RMP. So remember, among three ions, that is sodium, potassium and chlorine, sodium is not that much useful because it's going to generate positive voltage. Potassium is going to be useful because it generates a negative voltage inside the membrane, and chlorine ion as of now is also useful because it can generate negative voltage inside the membrane. But in the later part we will get to know that instead of potassium and chlorine, it is the potassium which is primarily responsible for RMP. How? Let us see further. See, as of now, at least we know that which ion is going to generate what voltage inside the membrane. So, because the membrane is dynamic, it cannot say that, oh, only potassium ion, you only come inside the you only move outside the membrane. We don't, I am not allowing sodium, I'm not allowing chlorine. It cannot do in that sense. Every ion is going to move. And when every ion is going to move, every ion has a right to generate a charge. And what charge it will generate? Depending upon its movement and depending upon its voltage. Say sodium ion generates a positive charge. So hypothetically, let us see, suppose if sodium ion is moving, only sodium ion is moving. So how much charge does the sodium ion will generate? Let us see. We are considering a hypothetical situation where sodium is moving from ECF to ICF, from ECF to ICF. So when sodium ion is moving from ECF to ICF, when sodium ion is moving from ECF to ICF, what charge is going to bring inside? Positive. Now let us imagine only sodium ion is moving. It is moving from outside to inside. And what charge is developing inside? Positive. So by concentration gradient, yes, it's going to move from outside to inside. By concentration gradient, it is going to move from outside to inside. But you know what will happen now? Because it keeps on moving from outside to inside, a point will reach where there will be so much of positive voltage inside the membrane that this positive voltage will repel. It will cause repulsion of the sodium ion movement. They will push the sodium out. I'll repeat once again. See, sodium ion is moving. Okay, sodium ion is moving from inside to from outside to inside, and it is developing a positive voltage. After some time, there will be so much of positive voltage on one side of the membrane that even though the concentration has not been disturbed, concentration wise sodium wants to come inside. Concentration wise sodium wants to come inside from outside to inside. But the voltage inside the membrane is so positive that is not allowing the sodium ion to move inside. So concentration will push the sodium in, but voltage will push the sodium out. So a point will reach where concentration power and the electrical power will match. This is known as electrochemical equivalence. This is known as electrochemical equivalence. So concentration gradient, remember, is balanced by electrical gradient. All right. So this is known as electrochemical equivalence. Electrochemical equivalence. And this electrochemical equivalence, remember, this electrochemical equivalence is a voltage which is dependent on the concentration. A voltage which is dependent on concentration gradient. We also call it as Nernst potential. We also call it as Nernst potential. So let us just see. All right, we have some issues here. So what we'll do is we will just wait for a second and we'll come back with this concept of Nernst potential. Till then, just wait. We'll go with the concept of Nernst equation in our next session. Thank you.